Running in progress. All right, chat, feel free to unmute, or typing into the text chat might be more useful. We just started recording.
Zoom people, can you hear us?
Yes.
Oh, are you? First, you wouldn't think that. You didn't even touch somebody. All right.
Why are these ports all awful?
Well, this building's.
No.
Are these for everyone?
No, that is your question.
Yeah, I don't have that many questions though. Okay. God. You're good, man. I need to.
Hey, language.
All right, we're good to go.
Hey, you're right.
All right, friends, we're gonna get it going. It's a school night, so let's get it going. All right, friends. So, if you answer a question, volunteering questions, you get a cookie of your choice. You either get a wow, some serious options. Yeah, sorry, Zoom people. It's still up. Okay. Okay. The exam this Thursday. Please get a good night's of sleep. Don't forget your cheat sheet. It'll focus on microtubules, cell cycle, and some pathways. They're concepts from week 6 through week 9, or topics 4 through 7. Yeah, check out the YouTube channel. There's also a lot of fun videos on YouTube that aren't done by us, but they're animated, so they're much better. Okay. You want to do this one? I didn't write this one.
Okay, these are tips from past TAs on how to study, using the quizzes to review, and not just going through the multiple choice questions, but actually thinking about how Laura could ask that in a potential open response. Find people to study with. That's always super helpful. I think that's one of the most important things, to talk through everything. Explain things to each other, and then utilize all of your practice questions. Specifically, I think the problem sets are probably the most helpful for that, and just try to engage in active studying. So not just rereading the material, but making sure you're actually actively engaging with the material.
All right, some continued advice. Active studying could be doing practice problems. Using the problem sets as like a baseline and making up your own practice problems is a great way to study. You could also ask your friends, what would happen if this mutation were to occur. Rewriting your notes by hand, I disagree with, but if that works for you, go for it.
Write it by hand.
Yes, do your note cards by hand, but I wouldn't rewrite your whole notes because that'd be a lot. But yeah, go to the YouTube channel. That's a great resource. Yeah, we can move forward through these things. Double-sided. This will be sent out. So if you need it, this is just the final exam substitution policy. Don't get too upset if things don't go your way on this exam. There will be times to make it up, and the final can do just that. This is just some math that was clearly done by an elementary schooler. And stop, take a deep breath. I think this was, yeah, you needed that. Okay. Mechanisms of gene expression regulation. So we got prokaryotic RNA polymerases, which use sigma factors, and eukaryotic RNA polymerases, which use basal transcription factors. And so these regulatory transcription factors control gene expression, and they're gene specific, meaning they're looking for a specific sequence and they're going to go there. They don't just bind with anyone. And so positive regulatory transcription factors are going to enhance transcription, while negative will suppress. Yeah, just one slide, buddy. Sigma 70, it helps RNA polymerase bind. This is in prokaryotes or eukaryotes? Okay. Have a cookie. Yeah. Okay. You get half a cookie. No, but that was rapid. I asked the question and it was there. Public shame of answering the question, right? Okay.
No, I was one, I was. Yeah.
All right. So sigma 70 has a consensus sequence, so it can regulate many genes. So what is a consensus sequence? It's the most common nucleotide at each position. So here's some graphics to describe it. In the left one, there's T, which has an 82% chance of being there, and that would be its consensus nucleotide. That would be the one that's found the most. All right, tryptophan operon. So an operon is a series of genes transcribed at the same time and controlled by the same promoter. Here's a little TRP operon in the top right. Some useful terms to write on your note cards. The promoter is where your RNA polymerase will bind. The operator is where the repressors bind. And inducible means the operon is usually off and it needs something to turn it on, whereas the repressible is the opposite. So it's usually on and it needs something to turn off. And that's our lovely TRP operon is a repressible because it is usually on and it needs to be turned off. All right, so with low TRP levels, there's no access to TRP, and so the cells need to make more of it. So these genes are going to be on. Whereas when there are high levels of TRP, there's abundant access to TRP, so there's no need to make any more. So the genes need to be turned off because if a cell has enough of it, it does not need excess. It's like if you eat too many vitamins, they'll just all go straight through you because your body doesn't need all of them. All right, here's a nice graph. This might be a nice note card graph if you're comfortable explaining all of it. And again, these slides will be sent out later tonight. So attenuation. This is the sequence before the genes. So if these are all our genes, the attenuation sequence is right here. What happened?
No, nothing. I was just trying to gesture for this.
Oh, I like that. We should use a laser pointer. All right. So the 3 hairpin loops are the 1 and the 2, the 2 and the 3, and the 3 and a 4. And we'll go through what all those mean. So when there are high levels of TRP, TRP binds to the repressor, and the repressor is then bound to the operator. So there's no initiation of transcription. Whereas at low levels of TRP, the TRP is not bound to the repressor, so repressor is not bound to the operator, and then full transcription and translation can happen, as shown with these little lovely blobs. So the mechanism when there are low levels of TRP is that the 1 and the 2 pause, and the ribosome catches up, because remember, this can happen simultaneously because we're in prokaryotes. So transcription and translation are happening at the same time. The 1 and the 2 pause, so the ribosome can catch up to the RNA polymerase. And so then the ribosome gets stuck in region 1. And because there's no TRP available, because again, we're at low levels. And so the 2 and the 3 form a loop. And that means that there's no attenuation because there's low levels of TRP. And so when the 2 and the 3 hairpin form, it's the anti-termination signal. This is going to happen. It's going to say continue. And so transcription and translation continue. And then there's a full mRNA transcript produced by the operon. On the other hand, what if there's low to high? What if you initially had low and you get to high? So same exact mechanism as the low levels, the transcription is going to start. But the 1 and the 2 make the pause, similar to before. That will always happen. But then the ribosome can move past those TRP codons, those UGG and UGG, because there's abundant TRP levels. So the ribosome doesn't get stuck, and it reaches the stop codon in region 2, and it sits there. And remember, when there were low levels, the 2 and the 3 formed a hairpin. But now that the ribosome is kind of sitting on top of it, the 2 and the 3 can't form a hairpin. So instead, the 3 and the 4 form a hairpin, and that's the termination signal. And so the RNA polymerase dissociates, and then transcription and translation stop. And so this results in just this little baby mRNA being formed. Not very impressive. Okay, any questions on TRP operon? Yeah.
Yeah, so
So will the ribosome always stop at that first region? Like 1 and 2 will always
Yeah.
bind, the ribosome will always stop, and then, so what allows? Do you mind explaining again, like
Yeah.
what happens from that point?
For low levels or low to high? Both?
So say there were low levels, right?
Yeah.
You want it to continue.
Yeah.
What happens when you want it to keep going?
When you want it to keep going, the cells have evolutionarily done this, where you see how this is getting stuck at the UGG. UGG codes for the amino acid of tryptophan. And because there are low levels in the cell, there isn't any that can come in and be added to the polypeptide chain. So it gets stuck there. And that, because it's stuck in region 1, this blue is associated with region 1, that allows this 2 and 3 to form. Whereas when there's high levels, this ribosome can just skirt past that UGG, and it can end on this stop codon, and it sits there until it finds, until the next sequence happens. And once it's sitting there, it's what sequence is it sitting on?
UGA.
Yeah, UGA, and it's right next to this pink one. There's a little overlap. So this can't rise up and form the 2 and the 3 anymore. And that's why the 3 and the 4 kind of form. And that's what's saying, oh no, initially we didn't have enough PRP, but now based on this, we know that we have enough.
Yeah.
So that's the way it works.
Can I add something?
Yeah.
Yeah, so Laura in the beginning of the semester, kind of emphasized this thing where like, so the proteins in the cell don't have, or hormones also, but in this case, I mean, they don't have eyes, but they don't see like where they're going or anything like that. There are chaperone proteins, but in this case, we're not necessarily talking about that. So what happens is when abundant levels of tryptophan are present, it is more likely for you to have the repressor binding onto TRP. It's not that like necessarily the amino acid is like, oh my God, I need to be there. It's like, oh, there is a higher chance of it bumping into the repressor, which then binds to it. So that's how like the abundant level of TRP prevents more synthesis of TRP. I don't know if that makes sense.
Anyone other comments, questions, concerns? All right, so here's some helpful YouTube videos. The top right one is a 10-minute one, and the rest are like 2 minutes. And that's the one I used to study a year ago. So feel free to check those out. Oh, it's gonna start them, isn't it? That's great. Okay. Is that? Yeah, okay. This might be helpful on your note card, more of like a summary. It could be helpful, it could not. Whatever would help you best remember would be, it's the best way to do it. All right, non-coding RNAs that regulate gene expression. So RNAi is involved in the degradation or suppression of mRNA translation. All right, first we'll talk about risk. Oh, never mind. What does risk stand for?
RNA do silencing complex.
Do you want a cookie? No, you should.
Yummy.
You know, I feel like we don't have enough questions. Oh, okay. So we're not gonna do any more freebies. I thought I had done the little translation. Okay. All right. So risk plus AGO or Argonaut. Argonaut is the enzymatic component, and it helps with mRNA degradation, whereas risk is kind of the protein that helps all this function. So risk binds to a guide RNA strand, and that targets another RNA through base pairing. And risk needs double-stranded RNA to function properly. All right, siRNA and miRNA. This is very important to know the differences between the two, and we'll have a few slides that'll hopefully get it down. So siRNA stands for small interfering RNAs. They come from exogenous sources, so they can be like viruses, anything that comes from outside the body. They're cleaved by dicer in the cytoplasm, and they lead to mRNA degradation, whereas miRNAs come from endogenous sources, so usually they're translated in, by your nucleus inside your body. They're cleaved by drosha in the nucleus and dicer in the cytoplasm, and transcription and translation from the genome, so the nucleus, so same as endogenous, and they lead to the repression of translation. I can't because this seems simply, but what does it say on that slide? I'm sorry, you're not on that one.
I don't know.
Okay, what is it? One? and mRNA degradation sometimes. Here are the two models. This is the siRNA pathway. It's not too important that you understand the whole mechanism. I definitely write it down on the note card. But I think knowing the differences between the two would be more valuable. Here's mRNA. I'm sure you saw this in Laura's lecture. And here's a good note card, kind of like differences between them, each line. So endogenous, exogenous, starts in the nucleus, starts in the cytoplasm, you know, go down the list. And yeah. Here's another model of the two pathways where there are parallels and where there aren't parallels. Is this your slides?
Yeah.
Okay.
There was a question in the chat. So someone asked, is this, sorry, I didn't see that, but they asked, is this dependent on TRP tRNA levels or just TRP levels? I'm assuming that's asking about the tryptophan operon attenuation.
It's tRNA. Because when the ribosome is, you can't really see it on this. If you look closer, you can. There's a little line, and that shows the polypeptide, the amino acid sequence being built. And so when it goes over this sequence right here, the ribosome is making that protein and it needs those amino acids, and the tRNA will come in and provide those. So it is tRNA, not tryptophan amino acids.
Yeah. Okay.
Okay.
Nope. Oh yeah, sorry.
Oh, no.
Okay,
Do you want-
I'll just.
Okay. Do you want to switch seats?
No, it's fine. Okay, so now we're going to go into our cell signaling pathways. First, just an overview of the types of signals that we have. So you can either have hydrophobic or hydrophilic. Signals are usually small molecules or proteins. Hydrophobic ones can pass through the membrane. They bind to cytosolic receptors. Those would be your sex hormones, cortisol, etc. We focus on the hydrophilic cell signals because they can't pass through the membrane, have to bind to a cell surface receptor. And even if it's small in charge, because of that charge, it can't pass through the hydrophobic lipid bilayer. So first we're going to go into the RTK signaling pathway. So the general mechanism for that is that you have your signal bind, which causes dimerization of the RTK receptors. Then this allows the intracellular kinase domains, which are part of the RTK receptor, they become active and they cross-phosphorylate each other. So this is happening on the tyrosine domains that are part of the RTK receptor on the intracellular portion. Once the RTK is activated, this allows for binding sites for other intracellular signaling proteins. So, yes.
Dimerization, is that just when the two separate tyrosine kinase domains come together?
Yes. Yeah. So that would be like before cross-phosphorylation happens. Yeah. It's always good to like think about them in terms of chronological order. I find is really helpful. So once the cross-phosphorylation has occurred, this creates those binding sites for other proteins. So an example of this would be your GRAB2, which binds to the phosphotyrosines, specifically through its SH2 domain, because that will bind to a phosphorylated tyrosine. GRAB2 will recruit your RAS activating protein, or SOS. And this will, so your RAS activating protein, that's kind of just diffusing around the cytosol by binding to the GRAB2 protein through its SH3 domain, which binds to the PXXP motif on the SOS. This localizes it to the cell membrane because your RAS is anchored to the cell membrane via a lipid. So your RAS activating protein isn't going to just happen to bump into RAS because it's diffusing around the cytosol. But having GRAB2 there will localize your SOS to the lipid membrane and therefore bringing it closer to RAS so that it can activate RAS. So the RAS activating protein will activate RAS by switching out the GDP and allowing for GTP to diffuse into the binding site. RAS is active when it's bound to GTP, not GDP. That's because it's a GTPase, so its intrinsic enzymatic activity is to hydrolyze GTP to GDP. So once the RAS activating protein allows for GDP to switch out and GTP to switch in, RAS is active, and this leads us to the MAPK pathway. So this is a kinase cascade. The active RAS will somehow activate MAPK3. We don't know how, but a key here is that RAS is not a kinase. It will not phosphorylate MAPK3. We don't know how it activates MAPK3, but not through phosphorylation. RAS is not a kinase. And then after that, MAPK3, which is a kinase, will phosphorylate MAPK2. MAPK2 will phosphorylate MAPK1. MAPK1 will go and phosphorylate other proteins, which will lead to changes in protein activity or activate transcription factors. And this will ultimately usually lead to cell division. And then at the end of the pathway, at some point, RAS will be deactivated, and that will have a gap, which is a GTPase activating protein, will help activate that intrinsic catalytic activity of RAS. It will hydrolyze its GTP to GDP and become inactive. This is a summary of everything that I just said. Hopefully, we feel somewhat comfortable. Are there any questions on that so far? Okay. It's kind of just like everything that I just said reworded, basically. So here are just some practice questions for us all. Laura really likes what if questions. She likes to ask hypotheticals to make you think about how things would change in a pathway if certain conditions were happening. So can you guys give me some examples on how you would stop signal transduction in this pathway, which means stopping the cell response, preventing cell growth or division, and shutting down the pathway? What are some ways that you could do that in this pathway? Yeah. Okay, yeah, so like have a mutation in RTK. And what would that do specifically? Okay, yeah. So on the extracellular portion, if you have a mutation there, signal can't bind. Yeah, absolutely. You could have you already had a cookie.
All right, get 3 more, you can have a cookie.
Okay. I'm not trying to be the cookie police. I just feel like they're being too generous, so I have to be the bad cop. I'm sorry.
They're generous. They were well deserved.
Okay, all right. All right, someone said mutate the tyrosine on the RTK into an alanine so that the SH2 domain cannot bind. Grab 2 with SH2 domain cannot bind. Yeah. Zoom person, you would get a cookie if you were here. They said that if you mutate the tyrosines to an alanine, grab 2 can't bind to that because grab 2 through its SH2 domain has to bind to a phosphotyrosine. Yeah.
Do you mind explaining the SH2?
Yeah, so a domain is just an independently folded portion of a polypeptide. It's not a separate subunit. But these specific domains, they have binding affinity for a phosphorylated tyrosine. That's all you need to know about it. Yeah. And you need to know like which specific molecule has an SH2 versus SH3. So that leads into the next question. We can go into that. So where is the SH2 domain in this pathway? Which molecule? Yeah. Yep. Would you like a cookie?
You're too generous, guy.
Is it the adapter protein that when it's binding to the activated RTK through the SH2 domain, or is it a RAS activated protein that has?
It's the adapter protein. So yeah, because the adapter protein is what's binding to the phosphotyrosine, and that's what the SH2 is attracted to. Oh, the person over Zoom said that they would happily grab the cookie tomorrow.
Oh, I think they're making.
I don't think I'm sorry. Yeah. That was a unanimous no. Okay. So I know I went through it pretty quickly, but can anyone tell me which molecule has the SH3 domain?
Cold call. That's not happening.
So an SH3 domain, I mean, this is gonna give away both answers.
cookie, guys. Lisa?
So no, but so the SH3, yeah,
Yes, yes. What do they form? What do they form? What's it called?
Phosphorylation.
So the SH3, yes, so the SH3 domain is basically a site where you're able to bind PXXP motif. And then they couple together. Oh, awesome. Yep, we lost. Okay, it's back. So basically, all the adapter proteins therefore is for you to be able to get the phosphorylation signal and like continue along with it through the RAS activating protein. And so that is necessary because you can't do that straight from having the phosphate group in there because you don't have an SH2, which is I forget the name of it. What's the name of the actual SH2 domain? It has a name for it.
Oh, I don't remember.
Hold on.
They don't need to know though. So
Well, but the technicality of it is basically the SH2 domain is binds onto a phosphorylated tyrosine.
yes, yeah.
Yeah, so it's like a whatever the name of
It's
that
like a phosphotyrosine something. or
binding
other.
domain.
Yeah.
Yes, and so that is why the SH2 domain is there, and then the SH3 domain binds to the PXXP motif, and then they couple together.
Yeah. So the PXXP motif is on the RAS activating protein, or SOS is also how Laura would refer to it. So that gives away the next one. Yes, good job, Lisa.
Ask it.
Asking
Yeah.
regardless.
Not worth a cookie though, because we gave
Okay. So now that we just
Everyone
talked
should.
about that, which molecule has the PXXP motif? No, the adaptive protein binds to a PXXP motif. Yes.
Do you want
yeah.
a cookie? Grab it.
Nice.
You're too generous.
This is so a number of cookies.
You're too nice.
Okay. So what are some ways that we could inactivate RAS? Yeah. Yeah, that's it. Yeah, he answered. So can you explain why? Exactly. Yeah, so you could inactivate RAS by having your gap there. If you had a gain of function mutation, well, this will answer part of the next one, but if you had a gain of function mutation in your gap, that would, cause you to have inactive RAS. So what is the gap and what is the GEF of the pathway? So, sorry, I think I misspoke when I said that. The RAS activating protein is not the gap, that's the GEF.
Oh, my fault.
Yeah.
I think that's three.
Three.
That's three.
Yeah.
That's three.
I'll give it to him. I'll give it to him. Yeah, you can have it. You can have it. Okay, so just to be clear, the RAS activating protein acts as a GEF because it's a guanine exchange factor. I think that knowing what these acronyms stand for, is really helpful, because guanine exchange factor, what is it doing? It's exchanging a guanine nucleotide for a different one. So you're exchanging your GDP for GTP. What is a gap? A gap is a GTPase activating protein. If you're activating a GTPase, you're hydrolyzing GTP to GDP. So what is the gap of this pathway? It's not in the diagram. Yeah.
Oh, well, I was gonna say the activated RAS protein, like the RAS itself.
No, because the RAS is what has the intrinsic catalytic activity. She mentioned it in class. It's called the RGS. It's just the gap in this pathway.
So does the RGS bind to the RAS protein and then allow the exchange for GDP to GDP?
Yes, in this case, it's not an exchange, you're just hydrolyzing GTP to GDP, so you're like causing that reaction to occur, getting rid of the phosphate. Someone said, which molecule has a PXXP motif? That is your RAS activating protein, which binds to the SH3 domain on the GRAB2. And then someone said, wouldn't the GEF be the RAS? So the GEF is not the RAS because RAS has a higher binding affinity for GDP, so it's not able to release GDP on its own. It needs another molecule to exchange GDP, release it from the binding site, and then allow GTP to diffuse in. Okay.
Fantastic.
Gain of function, loss of function, basically, if you had like a gain of function in your gap, you're going to constantly be hydrolyzing GTP to GDP. Your RAS will be inactive. Opposite with loss of function, you know, if anyone has any questions on those, but I think we can move forward. Okay, so now the cell survival signaling pathway. The default setting for the cell is to die. And then if you activate this pathway, the cell will not die. So in cancer, we get an overproliferative cell division phenotype, which means that your cells are dividing too much. And that's how you get tumor accumulation. And this could possibly be the consequence of an inactive cell survival signaling pathway or a hyperactive cell division pathway. Both a hyperactive cell survival signaling pathway and cell division pathway. Okay.
You're rocking it.
Thanks. Okay, so in cell survival signaling, first we have RTK dimerization and phosphorylation, same as what we just went over. And then you have your PI3 kinase, which binds to the active RTK. And then PI3 kinase, because it's a kinase, it has phosphorylation activity. It phosphorylates your inositol phospholipid. The inositol phospholipid is anchored in the lipid membrane. So in the triphosphate state, this is when PIP3 is activated. So it starts being diphosphorylated and then it gets phosphorylated to PIP3. And also on this slide is that P10 is a phosphatase, which would remove a phosphate from PIP3. So if you're going from PIP3 back to PIP2, that's inactivating PIP3 because it needs the 3 phosphates in order to be activated. So once you have an activated PIP3, so it's been phosphorylated 3 times, AKT binds to PIP3 through the pH domain. And then AKT needs to be phosphorylated by 2 other kinases. So it's not being phosphorylated by PIP3, it's just binding to PIP3 through that binding domain. And then it gets phosphorylated first by protein kinase 1, which causes just a conformation change, which allows it to then be phosphorylated for a second time by protein kinase 2. PK2 is also called mTOR as well. So the activated AKT, so it's been phosphorylated twice, it dissociates from PIP3, floats through the cytosol, it's activated, and this allows it to form its function, which would be phosphorylating BAD. And once BAD has been phosphorylated, so I think sometimes they associate phosphorylation with activating a protein, but in some cases that's not true. So in this case, phosphorylation is deactivating BAD. If you inhibit BAD by phosphorylating it, BCL2 is now able to be active and inhibit cell death. And I have another diagram describing both of these scenarios. So in scenario 1, you have your BAD not phosphorylated, it's bound to BCL2. BCL2 is an inhibitory protein. It inhibits cell death. Your inhibitory protein is being inhibited, so it can't prevent cell death. This leads to apoptosis. In scenario 2, you have your active AKT because it's been phosphorylated first by PK1, then by PK2. AKT is active. AKT phosphorylates bad, inactivating bad. BCL2 dissociates and is able to inhibit apoptosis. This is a summary of everything that I just said, but in words, if you're more of a reader, and all of that will be posted. Are there any questions on that? This is always helpful. It's at the bottom here, just showing like the different domains. So again, the SH2 domain, your PI3 kinase needs to be able to bind to the RTK, needs to be able to bind to those phosphotyrosines, same as GRAB2, that's how they bind. There are also 2 of these on AKT, because it needs to bind to protein kinase 1 and 2. And then there's a pH domain on AKT, which allows it to bind PIP3 and on PK1 as well. All right, so now the JAK-STAT pathway. The example that we use for the JAK-STAT pathway is for proteins that are involved in breast milk. So you have prolactin receptors. These are not RTKs. They do not have kinase activity. They are different from RTKs. They are not RTKs. So instead, you have proteins that are attached to the sides of the receptors, and these are your JAKs. They're just called just another kinase because they have kinase activity, not the receptor. So your JAK1 and 2 are bound to the receptor. Similar to an RTK, you need to have your receptor dimerize once the signal binds. And then the JAKs cross-phosphorylate each other. And then this activates the JAKs because they're phosphorylated. And then, did you have a question? Sorry. Oh, no, sorry. I don't know if you were. Oh, okay. And then the JAKs, they cross-phosphorylate each other, and then they cross-phosphorylate the receptor as well. This activates the receptor, the STATs bind to your phosphorylated receptor, and then this causes a conformational shift. They get phosphorylated by the STATs, or sorry, by the JAKs. These STATs then form a dimer, they enter the nucleus and act as a transcription factor. Okay, so in this case, by happy ending, they mean like having an outcome where signal is fully transduced to the cell. So how could we potentially stop this pathway or like shut it down? Yeah.
Make it so like the receptor can't be phosphorylated.
Yeah, exactly. So what type of mutation would that be? You're right, but if Laura were to ask this on an exam, you'd have to say.
Loss of function.
In what protein though, or what enzyme? What is phosphorylating the receptors? Yeah, loss of function of JAKs. Would you like a cookie? No. Cool. Someone had a question in the chat. Do they cross-phosphorylate the receptors, or do they phosphorylate the receptor they are bound to? They cross-phosphorylate. Or sorry, no, they phosphorylate the receptor that they're bound to. They cross-phosphorylate each other. Good question. Yes, the one that they're bound to. Someone else said prevent the JAKs from dimerizing. So you're not preventing the JAKs from dimerizing, you would be preventing the receptor from dimerizing, and that would also stop the pathway from happening. In this pathway, where is the SH2 domain? Yeah.
On the receptor that JAK phosphorylates.
So the receptor that JAK phosphorylates has a phosphotyrosine, which is what an SH2 domain binds to.
Oh, then it'd have to be on the STATs.
Do you want a cookie?
You sure?
Yes, thank you.
Okay. Would the receptor used in this pathway be an example of an RTK?
No.
No, that's correct.
Why would it not be an example of an RTK?
Because so RTK is receptor tyrosine kinase. The receptor itself is a kinase. In this case, the receptor is not phosphorylating itself.
Oh.
It has to be the JAKs.
So,
And then this is kind of a throwback to exam 1 material, but how would you get stat, the dimer, into the nucleus?
endocytosis. Oh, wait, no.
Yeah.
An LS?
Yeah, NLS, and what
What-
protein would get you
Yeah.
Important.
there? Do you want another cookie?
No, okay.
Cool. Good job. All right, so this is a summary of everything that I just said. Just an overview of domains. We already kind of went over this, so I'll be pretty quick. They're independently folded portions of a polypeptide that have a specific function. SH2 domains, they bind to phosphotyrosines. They are not on the RTK. They are what allows another protein to bind to the RTK. So for example, GRAB2 or STAT have SH2 domains because they bind to phosphotyrosines. The pH domain, binding site on a protein where triphosphorylated inositol phospholipid can bind to. So this would be HIP3 binding to AKT. And then your SH3 domain is a binding site for the PXXP motif. And that example would be how the RAS activating protein or SOS binds to GRAB2, which is your adapter protein. Okay, one more cell signaling pathway to go. GPCRs, they're the most abundant receptor that we have in our body. It's a 7-transmembrane receptor bound to a G protein that has 3 subunits, your alpha, beta, and gamma. In the inactive state, your G alpha is bound to GDP. When you have a signal or a ligand that binds to GPCR, this causes a conformational shift on the intracellular portion of the receptor, and that causes G alpha to be to let go of its GDP. So conformational shift in G alpha as well. It releases GDP. GTP diffuses in, G alpha is becoming active now. It dissociates from the beta gamma complex, which stay together. They can go diffuse off laterally along the cell membrane, do their own thing. G alpha will then go and start a signal transduction cascade. Deactivating G alpha occurs when RGS binds to it. Oh, that's my bad, guys. I'm sorry. I said RGS earlier with the RAS pathway. This is where RGS comes into play. Please disregard. That's my bad. So RGS in this case will accelerate your G alpha's GTPase activity and cause it to hydrolyze GTP to GDP. And then once deactivated, the G alpha will form its complex again with G beta gamma and then with the GPCR as well. So what is your GEF in the GPCR signaling pathway? As a hint, GEF stands for guanine nucleotide exchange factor. So you're exchanging guanines, yeah. RGS, it's not the RGS. Yeah. Okay. Yeah.
The GPC.
Yeah, exactly. So your GPCR, the receptor itself, is what's causing G alpha to release GDP and allow GTP to diffuse in. So once signal binds, that conformational shift occurs and allows G alpha to let go of GDP. So now what is the gap in GPCR signal?
RGS.
Yes, yeah, exactly. So because RGS activates your intrinsic catalytic activity. Common misconceptions. They are not enzymes. They do not have any intrinsic catalytic activity themselves. They are just, like for example, the GAP is just helping to activate your GTPase. So GEF again promotes the switch from GDP to GTP. GAP promotes hydrolyzing GTP to GDP. Yeah, and we've gone over all of those things. Okay.
All right, on to some microtubules. Does anyone have any lingering questions before we move on to the next section? Does anyone want a cookie for fun?
Someone has to get a terrible man.
Participate. How's that participation?
Zachary in the chat. Zachary, if we could give you a cookie, we would.
There's so many. All right, we'll ask some, my questions are hard. All right.
The nucleoside is a nucleotide without the phosphate.
Okay, spindles. M phase. Microtubules form spindle and separate chromosomes. So the types of microtubules we'll go into is the aster, which attaches to the cell cortex, which is just its like cell membrane part. Kinetochore, what? Attaches to kinetochores. We'll have an image, and interpolar overlap at the spindle from opposite centrosomes. And all of these microtubules grow from centrosomes. So here's a nice little image. I'm sure you've seen it in lecture. In the green, we have our aster. In blue, we have kinetochore. And pinkish red, we have interpolar. Oh, what are those black dots in the middle between the interpolar microtubules? Go through it.
Centromere.
Close.
Yeah.
It's
Centrosome.
what walks? Yeah, motor proteins. Those are a little. All right. Tubulin is a monomer of microtubules, and it is a GTPase, so it has enzymatic activity. The alpha portion has a GDP binding pocket, but the GTP is buried in it, so it doesn't really see the light of day. Beta, on the other hand, is exposed, and only beta can hydrolyze GTP. I don't know if that's right, but we'll go with it. It was on the lecture notes. So the minus end is the exposed end of the alpha subunit, and it doesn't have much growth, whereas the plus end is the exposed end, and it results in more growth. Yeah.
I thought it was like, it's just gonna be on the beta side, but I feel like the
Yeah.
alpha could.
I think you're right. Yeah, I think so. We'll go through the growth and degrowth. Degrowth. I think I just made a new word. All right. Here's the betas in green and the alphas in blue, and these are the dimers. And as you can see, the GTP is it's encapsulated, so not much hydrolysis will occur, but the beta is exposed. So the structure, it's heterodimers, as we saw in the last slide. There's an alpha and beta component. They're polar, meaning they have a positive and negative end. And so tubulin addition can occur at either end, but it's significantly faster at the plus end. Other structural elements, it's a hollow tube, 13 protofilaments around the circumference of the tube, and they all originate in the centrosome, where the minus end is embedded in a MTOC, in the microtubule organizing center, and the plus end extend from there. So here are just a few figures. This is a good representation of MPOCs, which are the yellow portions. Your little yellow dots, and the microtubules, the minus ends are there, and they're extending from there. All right, so some dynamics in vivo, which means in our cells, there's pretty much no growth at the minus ends because the attached nucleating sites are the gamma tubulin ring complexes, and so that's where they grow from. So there's not going to be addition once the minus ends are stuck. But the plus ends do grow and there's catastrophes where they split apart. So microtubule nucleation in vivo, so in our cells, the gamma tubulin ring is formed and acts as a base for the start of microtubule growth. This all occurs in the centrosome, or it occurs in the centrosome from, we go back to our little lovely, there's our centrosome, and we've got our MTOCs there. And the minus end, as stated, is embedded in these gamma tubulin ring complexes. It's kind of like a base to build up from. It's like your foundation in a house. You build up from there. So that's our minus end. Any question? All right. So dynamics in a lab. I can all but guarantee there will be one of these figures, or you'll have to draw one of these figures on the exam on Thursday. So make sure we'll have a few practice questions of exam type questions, so we can go through those. So the setup for a lab experiment is you take pure tubulin with some GTP in a test tube, and this is what the produced graph will look like. This is absorbance on the y-axis and time on the x-axis. The pros of doing this in a lab environment is that it's more controlled, but the cons is that what you find in a cellular in the lab environment might not be true in a cellular environment. And graph is absorbance versus time. So first stage is the lag phase. And so it takes a little bit of time for these microtubules to get started. The higher the number of free tubulin, the shorter the lag phase. And as you can imagine, if there are more MTOCs, some organizing centers, there will also be a shorter lag phase for it to get going. The next stage is the elongation phase, where the rapid addition of tubulin binding occurs. And as mentioned, this is primarily the plus ends because of that greater affinity. And as microtubules elongate, the concentration of free tubulin decreases. So eventually we reach that plateau where the slope decreases over time. And the final phase is the plateau phase, where the rate of microtubule assembly is equal to the rate of microtubule disassembly. This is not when they're static and the microtubules are just chilling. They're going up and down, but this is an average of that plateau. And it's when GTP hydrolysis catches up. And eventually in a lab environment, when there's no added GTP, eventually all the tubulin will be GDP bound because it will hydrolyze the GTP. All right, so this is, oh yeah, question?
Yeah, so if that's the case and all tubulin will eventually be GDP pound, that means that like when tubulin is GDP pound, the microtubule disassembles, right?
Yeah.
Yeah, so wouldn't this graph like
If it kept, yeah, if the x-axis kept going, you would see a decrease, which I think would be pretty helpful as well. But yeah, so yeah, you're right. All right, an exam type question. What if stable aggregates, so seeds, already existed in cells? What would happen? What phase would be quicker? Lag, exactly. Yeah, because there's already sites for those microtubules to bind and just start growing. And an oh, do you want a cookie?
Oh, no, I thought it was.
Oh, okay. In an in vitro assay, so we're in a lab. What happens after GTP hydrolysis catches up and there's no more free GTP? I think we just answered this, but they'll slowly dissociate in catastrophe. So here's a nice little sheet to maybe put on your note card. When GTP hydrolysis is greater than tubulin addition, there's shrinkage or depolymerization, and this is not a stable, and there's no GTP cap, whereas the inverse is true the other direction. Here's another figure of them growing and shrinking. The red little dots indicate a tubulin molecule with bound GTP, whereas the other ones don't. And I would write these down on your note card. I think, do you remember what our exam had? Was it Taxol?
Oh.
It was Taxol. I would write down, of all these drugs, write down Taxol. But it could be any of them. So, it's just a small little table to put in, and these slides will be sent out as mentioned. All right, so some MAPs, kinesins and dyneins. MAP is a microtubule associated protein. Tau is highly expressed in the central nervous system, in particular your neurons, and it binds and stabilizes microtubules. So it helps them build up during the M phase. And dysfunction results in neurodegeneration. So hyperphosphorylation of these tau proteins makes them not able to bind to microtubules, and thus the microtubules aren't really stable. And then this tau protein aggregates with itself and it forms these things called tangles. And so this is seen in Alzheimer's and some other neurodegenerative diseases. It's a very fascinating field. Motor proteins, as those black dots at the first diagram we showed for this section, they hydrolyze ATP to move along the microtubule. Pretend the microtubule is a highway and these are the cars. They're moving, they're transporting cargo, and they do have directionality. So a kinesin is a plus end directed, except for kinesin 14, that's the exception. And they're diverse. There's many functions of these kinesins. Dynein, on the other hand, is always going to be minus end directed, and it's very common in flagella and cilia. Is it cilia?
Cilia.
Cilia.
Yeah.
Cilia. Why didn't they spell it that way?
Best pronunciation.
No, you know what? I think I overcompensate. So spindle formation requires an array of opposing forces. So for example, egg 5, which binds to interpolar microtubules that overlap, is a plus end directed motor, but it's a special kind. And so what it does, oh, never mind, I thought I had a picture. It has dimers both on, it has feet both on both sides, so it'll walk towards the other ends and push the plus ends apart. There's definitely a video on YouTube, but egg 5 is one of the most important kinesins that we talk about, and I would expect an exam question on this kinesin.
On egg 5.
On egg 5. So how does egg 5 push these? I'm gonna put the this is a normal kinesin. You see it's got its little feet and it's moving towards the plus end. Egg 5, on the other hand, has feet on both sides, and it's going to help in that interpolar microtubules that overlap, and it's going to help them push apart. So it'll help them dissociate.
No.
The other way. It'll help them like grow apart.
Yeah, because it's walking towards the plus end of each. So wait, right?
It itself is
Yeah.
not moving. It itself is not moving. This red little protein isn't going to move, but it's going to push the pluses away from each other. Right. Oh no, push them towards each other.
No, your thought
No.
process is right, Scott.
Oh,
Your
yeah.
thought process is right.
Yeah, push them away because it's
Yeah.
walking towards the plus end. So it's like
So push-
pulling the top one that way, pulling the bottom one that way.
Yeah.
Pushing them apart.
But in itself is staying in that same position. These are types of kinesins that we might talk about. I don't know how important these will be on the exam, but if you have extra space on your note card, maybe write down their functions of each.
You need to know that kinesin 13 diffuses laterally and it causes depolarization. And then you should also know that kinesin 14 is a minus end.
Yeah, I think that was touched on earlier, but yeah. Here's another figure of them moving cargo in their directionality. Very cute. And here is a motor protein walking. They play some pretty dramatic music here, so it's nice. It's a nice little chuckle if you want to, you're studying and you need to see it. So, all right, last section. We're almost there, friends.
Killed it.
Does anyone want a cookie? We're getting to the late hours of the night. Come on, we got to finish them off.
All right.
Sing.
Take some home to your roommates.
I'll take some home to
Okay.
All right, so with the cell cycle, we're going to start talking about our CDKs. These are the master regulators of the cell cycle. Ooh. Okay, so CDKs are serine threonine kinases, which mean they phosphorylate on either a serine or a threonine. And they are required in order for the cell cycle to occur. They're regulated by cyclin, specifically like cyclin levels. So if you want to change the concentration of cyclin, that will change the activity level of your CDK. They're also regulated by phosphorylation or dephosphorylation. And this doesn't necessarily mean active or inactive, because you can have an activating phosphate group added, you can have an inactivating phosphate group added. And there are specific CDK inhibitors as well. So you need both the activating phosphorylation, which is done by your CDK activating protein kinase, and you also need cyclin in order for CDK to be completely active. And then anytime there's an important checkpoint during the cell cycle, the cell cycle will pause by inhibiting or deactivating your CDK. And again, CDK can't work unless it's bound to cyclin. So cyclins are required for CDK activity. They help activate CDK and tell CDK what to phosphorylate, because you have specific cyclins that are present during different phases of the cell cycle. And so they help direct CDK to specific substrates. Cyclin is regulated by its concentration. So you can either do this through regulating transcription and translation at specific points, or by degrading cyclin through ubiquitin-mediated proteolysis. And then cells produce the specific cyclin that they need for the specific cell cycle phase that they're corresponding to. So for example, you have different cyclins for different phases of the cell cycle. And then once it's not needed, it'll be degraded. So this goes into more detail than you really need, but essentially you need both the CDK to be bound to cyclin and you need CDK to be phosphorylated, but this happens in a specific order. You need cyclin to be bound first, and then your CDK activating kinase will add a phosphate group to CDK and allow it to be fully active. You can also have a CDK inhibitor, which will block CDK from phosphorylating its target protein or prevent it from binding to its substrate. So going into the different phases of the cell cycle, during G1 phase, Laura didn't go through all the phases during this first week of cell cycle, so we're just really going to focus on G1. So the cell is looking for if it's big enough, if it has enough energy, does it have all of the machinery in the cell, the proteins, the enzymes, is there any DNA damage? And then the G1 CDK complexes are not yet active. So when we say G1 CDK, that means that there's a cyclin bound, because we're referring to the specific cyclin that is specific to G1 phase. So what does the cell need in order to start thinking about cell division? What does it produce as a result of this? It needs cyclin, so that's what's being created. But what will help initiate cell division? This is something like referring to a different week of this same unit.
Oh,
Yeah. Exactly. Yeah. So it needs a signal to bind. And yeah, someone in Zoom said an external signal. So that would be like a growth factor that binds to the cell and will initiate cell division. And then it will produce G1 cyclin as a result. So in order for this to all get started, you need the free replication complex to form. And this is when CDK activity is low. So this is before CDK gets activated. First, you have ORC binding to the origin on DNA, and ORC is an ATPase. This will recruit CDC6, and that will recruit CDT1 and MCM, which is your helicase. And together, all of these proteins form the pre-RC. CDT1 leaves. It's not technically part of the pre-RC, but it's needed to help form this whole complex. They're all enzymes, all of the proteins that are part of the pre-RC, not CDT1. Okay, so after you have your pre-RC formed, you then have if there's DNA damage, this is when P53, which is a transcription factor that we'll talk about more, that's when P53 would get activated and prevent the cell cycle from going through. But if you don't detect DNA damage at this point, you reach the restriction point. This is the point of no return where the cell either commits to the cell cycle or not. So if everything is in check, your G1 cyclin CDKs become activated and they will phosphorylate RB. So in this diagram, you see that RB is bound to E2F. E2F. So if RB becomes phosphorylated, it will release E2F, and this will allow for transcription to occur. E2F is a transcription factor that will upregulate certain proteins that are needed for S phase. So it'll help push the cell into S phase out of G1. It's kept inhibited by RB until there are enough cyclin G1 CDK in the cell. So once G1 CDK inactivates RB, E2F can be activated and transcribe the proteins for S phase. Some of the proteins that get transcribed and translated are more CDKs, or sorry, cyclins that will bind to CDK and further phosphorylate RB. So this is a positive feedback loop. You can't stop this once it's happening. This is why it's the point of no return. And again, after this point, there's no going back. So why is P53 a tumor suppressor? Yeah, so it does both, right? So P53 is a transcription factor. It will cause transcription of P21, which is an inhibitor of CDK. If you don't have P53 in the cell, if it's no longer available, what will happen?
Cell division will always happen even when there's DNA damage.
Exactly, yeah. There's nothing to stop cell division from occurring, and so it'll keep happening. This would lead to a cancerous phenotype. So if P53 is suppressed or damaged, what happens? We kind of just answered this. You would have more cell division occurring. This would not be good for the cell. True or false? In response to DNA damage, cells should increase concentration of CDK inhibitors. Yeah, why? Yeah, exactly. CDK is what's pushing your cell through the cell cycle, so you want to stop them from doing that. True or false? In response to DNA damage, E2F is active. Right. And how come?
E2F is the one that like causes the pre-CD form, right?
So this is after the pre-RC has formed.
Oh, the pre-ILP, right? Something like that, like.
Yep, after that as well. So you have the pre-RC form, then the pre-IC, and then you're at your restriction point. So this is the point where E2F comes into play. It's a transcription factor. What is its role as a transcription factor?
Helps push the cell to divide.
Yeah, exactly. Keeps it going. So this would be false because if there's DNA damage, you're not going to pass the restriction checkpoint. E2F will be inactive. So could E2F be a cause of cancer? And can a cell divide if E2F does not function? We'll start with the first one first.
Yeah.
But it's not.
Yeah, exactly. Because again, the whole restriction checkpoint, we need E2F to be there. And then last one, true or false, pre-RCs can form at the end of G1 after CDK is fully activated.
False.
False, yeah. And how come?
Because pre-RCs, CDK helps form pre-ICs, and so if pre-RCs form at the end of G1, then it wouldn't be ready to go into like past the restriction point.
Yeah. Exactly. CDK prevents the formation of any new pre-RCs because you don't want all of this stuff occurring more than once in the cell at a time.
Actually, I know that there was a problem with this on the problem set. I don't understand why when G1 CDK are added prematurely, pre-ICs or pre-RCs don't form. So I know that like CDKs make the pre-RC into the pre-IC, but does the pre-RC have to like be there first?
Yeah, the pre-RC can only form if you have low levels of CDK, because once CDK is there, it inhibits formation of new pre-RCs.
Okay.
So yeah. Okay, and then just briefly, she doesn't really go into this that much, but it's all really connected. So during S phase, the cell is looking for DNA damage. P53 activation can happen at the beginning of this phase as well. And this is exactly what we just said. The S phase CDKs inhibit new pre-RCs from forming because you only want to replicate your DNA once. This is an overview.
So pretty.
It is very pretty. Yeah, we've gone over G1. G0 is when it's in a non-dividing state, and that would be like it's waiting for a growth factor to bind to a cell receptor. S phase is your DNA replication. G2 is when you have your second gap and it's preparing for chromosome segregation. And then M phase is when you have your actual chromosome segregation, and then you complete the cell cycle. Alrighty, folks, we made it.
Lingering questions. Lingering desires to have a cookie.
Actually, I have one question.
Yeah.
Yeah, the medicine portion or like the like drug treatment for like the like Taxol and all that stuff. You mind just going back to that really quick?
Yeah.
So how does Taxol bind and stabilizing microtubules? Is it because the microtubules can't then disassemble? That is why like it messes with the cell division.
That's a good question. Let's do some field research.
I'm sorry.
Yeah.
I think
So,
it's a catastrophe for movement, so like if you inject it, kind of freezes.
but is catastrophe required for cell division? Like,
No, catastrophe is a natural so the microtubules need to be like. They need to be stable, but they need to be unstable to the point where they can retract if necessary. Cause they're like actively, if you think about like, I'll give you the example for like spindle assembly, right?
Yeah.
If they're actively going towards the center of the cell, like if you think about the size of like a tubulin diameter compared to like the size of the actual cell, like you need a whole bunch of tubulins to be able to do that. And it's not because you're forming a microtubule that's long enough that you're actually going to be accurate to reach the kinetic port. And so like if you mess up, if the microtubule goes in the wrong direction, doesn't actually reach it, you need to like retract and go back until it actually lands. And if you think about like the amount of them that you need, like it kind of like makes sense as to why, like the stabilizing factor.
You need to both break down your microtubules in order for this spindle to form, and then during anaphase, you need your spindles to be able to disassemble to pull your chromosomes apart. So if you're stuck in anaphase, you can't pull them apart, your cell's gonna die.
So this is going really in depth. I don't know if we'll have to know for these yet, but if the plus end is the one that goes outside of the MTOC and it's the one that attaches onto the centromere on the chromosomes, right? Isn't it also the plus end is the one that is the plus end the one that depolymerizes first? Like the minus end doesn't depolymerize once in the MTOC, right? It's just the plus end.
Yeah, the plus end has that GTP cap, which kind of protects it. It's kind of like the 5 carbon cap, but eventually GTP hydrolysis over time will catch up, and that's when it starts the catastrophe.
It's from that side, and the microtubule is like being disassembled from that side. Then, like, just out of curiosity, do you know how it can still attach to the chromosome and bring it back when at the same time that's the side that's also disassembling first? Well, I'm just curious. It's not like a
No, you're actually gonna like kind of go into this in like the next week.
I was just curious.
Yeah, no, it's a good question.
It's a good question. I just don't remember.
Yeah.
I'm sorry. I will have an answer for you next week. Yeah.
there's like why is taxon an anti-cancer drug and
So did you want to answer?
Like, the stabilizing,
Yeah.
because if it's stabilized, then you can't do like dynamic stability, like it's like needed. So then it just leads to the cell going to like cell death.
Take a cookie, please.
Because like, if there's no stability, like the chromosome.
Yeah, there's
Goes
stuck in
the middle.
It's what we just, yeah, so it's basically what we were just saying, that like, if your microtubules are too stable, they can't have the dynamic instability of being able to also break down and reform. So if you get stuck in anaphase, your cell can't pass the spindle assembly checkpoint. It'll eventually go into apoptosis. And so for cancer cells, they then can't divide. Yeah. Yeah. Yeah.
Well, I think that's because, like, it's not so much that. Like, because of the exact form that won't necessarily trigger a checkpoint before that.
All right, dude, we're gonna stop the recording, but feel free to keep asking.